All welcome to another video from Life Science Library. In this video we are going to see about amphibian development Csirnet previous year question concept series 2008 to 2020. First question we are going to see about noggin. Noggin is a secretory protein by sperm and organizer in Xenopus. Noggin is produced to inhibit beta bone morphogenic protein and it helps in docilization. It induces to form neural cells, otherwise everything will become ventral. Plasmid cloning expression of noggin protein is microinjected into UV treated embryo which does not give rise to neural tube, it will rescue normally. This is a static part. Present in Gilbert textbook, you can verify. Amphibian development. With respect to amphibian development, in the dorsal side we have beta-catenin. Beta-catenin expression is being produced in the dorsal side. This beta-catenin will go and attach to TCF3 and will produce the expression of cymose and twin gene. And also give rise to guscoid. All occur in the dorsal side. This is the concept. So, neurope center is formed in the dorsal side of the embryo due to the accumulation of beta-catenin which helps to activate cymose and twin gene. Now with respect to activin. Activin is a morphogen. This activin, if it is produced in the very high concentration, it will form guscoid. If it is produced in the low concentration, it forms X-bra. This X-bra will go and inhibit guscoid. This is the whole concept. Beads without activin does not elicit the expression of either X-bra or guscoid, correct. Highest concentration activate guscoid, correct. Highest concentration induce X-bra, wrong. C is the wrong statement. Cell farther from the beads getting negligible activin activate X-bra gene and become blood vessels and heart. No. Cells farthest from the beads getting negligible activin activated either X-bra, neither X-bra or guscoid, and default gene expression is instructed, that is blood vessels and heart. So blood vessel and heart are default expression. Next, dominant negative concept. If GSK3, so we have seen the properties of GSK3. GSK3 will go and inhibit beta-catenin. If GSK3 is removed, then it will form secondary axis. No, now 2018. Which of the following statement with respect to amphibian development is correct? Organizer itself is induced by neurope center located in the dorsal most mesodermal cell. It is a wrong statement. Organizer functions by secreting proteins like chordin and organ polystatin that prevents BMP signal that could otherwise dorsalize the mesoderm. Wrong. In the presence of BMP activated, the ectodermal cell will form neural tissue. Wrong. So D is the correct statement. WAT signaling causes a gradient of beta-catenin along the anterior posterior axis of the neural tube and specify the regional cessation. So D is the right answer. – 2017. Dorsal-most vegetal cell of the amphibian embryo is capable of inducing organizer is called neurope center. Dorsal-most vegetal cell. So neurope center is a endodermal cell marked by the presence of beta-catenin. Next, Dominant negative question. It will form secondary axis. Okay. Noggin. We have seen about noggin. Noggin is not a transcription factor. A is not a right answer. Noggin induces ventral fate. No, noggin induces dorsal fate. B is not right answer. Noggin is involved in organizer fate. Correct. It's required to induce secondary axis. Correct. B is the right answer. With regard to organizer, sperm and manganol diagonizer. Sperm and manganol diagonizer. It induces host ventral tissues to change their fate to form neural tube and the dorsoventral tissues. Correct. It cannot organize host and donor tissues into secondary embryo. No, it can do it. So B is not right answer. It does not have the ability to self-differentiate dorsal mesoderm. No. C, not right answer. Ability to initiate movements of gastrulation. Correct. Both beta-catenin and cordon are produced by organizer. No, is not correct. A and D is the right answer. So amphibian dorsal ventral axis formation. Now if you see the concept of dorsal ventral axis formation in frog. So if the sperm enters into the egg in this side becomes ventral and the opposite side will become dorsal. There is a cortical rotation will occur at 30 degree. That forms grey crescent. Okay. So dorsal ventral axis is formed by the point of sperm entry. Next question, due to lens formation. Lens formation, optical vesicle, and consider this as a ectoderm. Anterior portion of the head ectoderm, and the signals produced by BMP signaling and FGF8 signaling produced by this optical vesicle induces head ectoderm of the anterior region to form lens. This is the concept. So let's see about in case of Xenopus. In case of Xenopus, optical vesicle induces the trunk ectoderm to form lens. Optical vesicle cannot directly do or induce, there are signalings. B is not correct. Abrupt is B correct, but technically B is not right. The lens induction can be achieved by the absence of optical vesicle. No, A is not correct. C and D is the right answer. Inducing organizer. Which can induce organizer? Neurobe center. Hansen node will be seen in the case of chick. So we are making a thing about Hansen node and this dorsal blastopore lip in case of frog. This occurs in case of chick. What are the similarities? It expresses the same marker gene. Both expresses same marker gene. They induce secondary axis, they induce gastrulation, all those things. So A and C is the right answer. FGF receptor. FGF mostly involved in the formation of trunk and tail. It has nothing to do with head. So C and D is wrong. Not only forelimb but also hind limb. So A is the right answer. This is a blank question that anybody can answer. See, amphibian due to some injury, 2013. Amphibian due to some injury, when the lens is damaged, it is fully differentiated into iris cells and can regenerate the lens. It is achieved through the following possible process. Iris cells through some signaling undergo de-differentiation. Correct. Same statement is repeated. De-differentiated and then conversion into lens cells. Correct. A and C is the right answer. 2013. Xenopus. 2012. So neurobe center, I said it is endodermal. It is vegetational and origin. It is endodermal. So B can't be the answer. D can't be the answer. Sperm and organizer. Mesodermal in origin. It's not ectodermal. A is the right answer. Primordial germ cells. Germ cells are endodermal. In case of frog, I am speaking in case of frog. It's mentioned as amphibian. So C is the right answer. Norgan. I have discussed about Norgan earlier. Norgan binds directly with BMP and BMP2 and thus prevents the signaling complex. D is the right answer. Norgan is a secreted protein that induces dorsal ectoderm to form neural tissue and it dorsalizes the mesodermal cell which could otherwise contribute to ventral mesoderm. A is the right answer. Norgan cell along with other molecules prevents BMP. It's not with the other molecule. It itself go and inhibit. So C can't be the right answer. B is the right answer. Amphibian. We have seen the same thing when we saw about cleavage. Amphibian is holoblastic and mesolecithal. A is the right answer. Experimental based question 2010. Lateral separation of amphibian embryo in 2-cell stage. Consider this is an amphibian embryo in 2-cell stage. If these two cells are isolated as the separate cell, then they will form two identical twins. Last question from GATE 2009. Maturation promoting factor. Maturation promoting factors. Leave it about amphibian, xenopus, whatever. What is the duty of maturation promoting factor? Regulate cell cycle. In general conditions, regulate cell cycle. Composed of cyclin and cyclic kinase. Yes, correct. It undergoes periodic degradation. Yes, correct. It undergoes periodic phosphorylation. Correct. So, D is the right answer. You don't need a knowledge about xenopus, frog, nothing else to answer this question. Simply if you know cell cycle, in case if you are strong in cell biology, you can handle this question. So, D is the right answer. So, in this video we have seen the concepts of activin, Norgan, cordon, follistatin, beta-catenin, goosegoid, twin gene, expra, then what else? Sperm and organizer, new root center, dorsal ventral axis, so far we have seen all these concepts, all the basic concepts and the cleavage of about frog. So far we have seen all the basic concepts from frog. Hope this video is very useful and you can gain so many knowledge from this previous year question concept study. Hope this thing is very helpful. You have better conceptual understanding in a short period of time when you go through previous year question. Hope this is very clear. This video is useful. Please do like, share and subscribe to our channel and refer this channel with your friends and fellow aspirants. Thank you for watching this video. Thank you.